Hello everyone and welcome to video 8. So you guys should be experts in proteins, so you should know all 5 different proteins and be able to state their functions. So if you don't know that, then you need to go back and watch video 7. So we're still on key area 4, so if you want to print out the activities and consumables, you have to go to 4 proteins and then you go to 4.2 enzyme structure. So same with the PowerPoint, it'll be 4.2. So for your starter activity, you need to name 2 proteins and state their function. For 2, you've got to say what an enzyme is. For 3, you've got to state the enzyme's function. And for 4, you've got to name an enzyme. So if you want to pause the video just now, and then I will reveal the answers to you. So the answers for the starter activity are, you could have put any of the 5 proteins, so you could have put enzyme and then its function would be to speed up chemical reactions. You could have put antibodies, which would be to protect the body against diseases. You could have put hormones, which are chemical messengers, or structural, which are components of frameworks like your protein channels. You could have put receptors, which receive signals and produce a response when molecules bind. So what are enzymes made of? Well, we should know that they're made up of proteins. And what is their function? So it's to speed up chemical reactions without being changed in living things. And then name an enzyme. So you've got amylase, lipase, catalase, protease, and pepsin, which we're going to look at in more detail. So don't worry if you don't know any of the names of the enzymes. You probably would have got the amylase one from your saliva or from bird year. Okay. So your learning intention today is to state the different properties of enzymes, make sure that you can write out the reaction equation for enzymes, know that enzymes are specific to only one substrate, and name the 2 different types of reactions enzymes are involved in and provide examples of each, and also know how enzymes are involved in industries. So to be successful, you need to be able to know and write down the equation for a reaction involving an enzyme. You need to explain how enzymes are specific, and you need to compare the difference between synthesis and degradation reactions. And you need to be able to answer questions about the different synthesis and degradation reactions that you get. So enzymes are extremely important. Without them, we would not be alive. So what we need to know is, what are enzymes, what do they do, and what happens to them after a reaction. So we need to make a note of this. So you would put the title enzymes. So your properties of your enzymes include they are produced by all living things, plants and animals. So they are made up of proteins and they are biological catalysts, which means they speed up chemical reactions without being used themselves. So they remain unchanged throughout the reaction. They're involved in 2 types of reactions, so degradation, so they can break down things, and synthesis, where they are involved in speeding up build-up reactions, so where things are built up, so substrates are built up. And enzymes are specific. They're also affected by pH and temperature, and we'll go into that in more detail in video 9. So our importance of enzymes are they are involved in life processes like respiration and photosynthesis. So these processes would not be able to continue without enzymes, as they enable biochemical reactions to proceed at lower temperatures, about 5 to 40 degrees Celsius. So that is why if you have got too hot of a temperature, your enzymes would not work properly, and that would lead to you becoming ill. And same with if it was too cold, your enzymes in your body would start to slow down, and your body would no longer function properly, and you would die in either instance if it was too hot or too cold. So you guys need to know the enzyme word equation. So the substance that an enzyme works on is called the substrate. So the substance that's at the very start of the reaction will be your substrate, and then the substance it makes is called the product. So your substance your substrate makes is called your product, and this would happen without your enzyme. It would just be a very, very slow process. But what your enzyme does is it speeds up the reaction, so it makes it go faster at lower temperatures. So you need to make sure that you write out this word equation and that you know it. So then you would want to put the title how enzymes work. So enzymes are made of protein, which we've already discussed, and this protein has a special shape with its which is unique to each enzyme. So enzymes have this part of them which is called the active site, and you guys need to become very familiar with this active site. Okay, an active site is a specific shape, so it will only fit specific substrate. So it will only fit one substrate. See how it's a triangle, and only the triangle will fit into the enzyme. If it was a square substrate, it wouldn't fit in. Or if it was a circle substrate, it wouldn't fit in. It's got to be that triangle shape, and it's got to be perfect so that it can fit into the enzyme. So the substrate always binds at the active site. So as we said, our enzyme only fits one substrate, so other substrates that are the wrong shape to fit in the active site of the enzyme. So this is described as being specific. So there's a video that shows you how enzymes work and how they are specific. So remember that only one substrate, which is a complementary shape, will be able to bind to your enzyme's active site and will be able to react. So complementary is a word that we need to use to describe this. So complementary. For your 2 marks, because usually you get asked how does your substrate and your enzyme work together, or how are they specific, and you would want to say that your substrate is a complementary shape to your enzyme's active site. So you'd get 1 mark for saying that they're complementary shapes or matching shapes, and the 2nd mark for pointing out that it's the active site that the substrate binds to. So complementary is the key word there. So we're wanting to summarise about enzyme active site specificity, so basically what we've already said. So an enzyme can only act on one type of substrate, so it's said to be specific, and the substrate molecule exactly fits the enzyme's active site, so that is the key word there, active site. And the shape of the enzyme's active site is said to be complementary to the substrate's molecular shape. And this is sometimes described as a lock and key, because only one key fits one lock, just like one substrate fits one enzyme's active site. It's complementary to the active site. So the shape of the substrate is complementary to the enzyme's active site. So then after that, the substrate will then create a product. So what we're going to do is we're going to look at an experiment just now, and we're going to look at the specificity of enzymes. So we're going to have 3 different enzymes, and we're going to add starch to each of our dimple trays, and then we're going to see what happens. Now we know, or we should know, that there is an enzyme called amylase, and it's found in our saliva, and it breaks down starch into glucose. And we know that we can test for starch with iodine, and when we test for starch with iodine, iodine is a brown substance, and if starch is present, it turns blue-black. If starch is not present, then it will go brown. So here we've got the experiment. So you've got starch in enzyme A in column A, and you've got starch in enzyme B in column B, and you've got starch in enzyme C in column C. So we're going to add a drop. So 3 test tubes were set up as shown above, and then samples were removed every 3 minutes and tested for starch. So at 3 minutes, you've got your sample and then iodine added, and this go to blue-black, saying that there's starch. After 6 minutes, iodine's added again, they go blue-black. 9 minutes, again they go blue-black, except from A stays slightly brown. And then after 12 minutes, in A it's brown, but in B and C they stay blue-black. So this suggests that starch only disappears in tube A. So the enzyme in this tube must be amylase, since only amylase can break down starch. So enzyme B can't break down starch, it doesn't speed up the reaction of starch to glucose. And enzyme C doesn't speed up the reaction of starch to glucose. Only enzyme A has had that effect. So only enzyme A, which is amylase, can speed up the reaction of breaking down starch to glucose, which we've seen when our starch disappears. So only amylase will have an active site that's a complementary shape to the starch. So you would want to write a note on your induced fit. So this is another mechanism of action as well as specificity that happens. So when a molecule of your substrate enters the active site, the shape of the enzyme molecule and its active site slightly change, making the active site fit closely around the substrate, which is called induced fit. This close contact increases the chance of a chemical reaction taking place and helps to speed up the chemical reaction. So here's a diagram of your induced fit that you'd want to put in. So you've got your substrate entering your active site of your enzyme, and then you've got your enzyme substrate complex. So your enzyme changes shape slightly as the substrate binds, and then you've got your enzyme product complex, and then you've got your products leaving your active site. So you would want to put the heading the types of enzyme reactions. And there's two types of enzyme reactions. So two types of reactions that enzymes can speed up. One of which is degradation reactions where your substrates are broken down, and the other one is your synthesis reactions where your substrates are built up. So here are some examples of degradation reactions. So we know that proteins are made up of amino acids, so we've got a long chain of amino acids. So what happens is the enzyme protease speeds up the reaction of proteins being broken down to amino acids. The same with our starch molecule that's made up of lots of glucose molecules. So our enzymes, carbohydrates and amylase, they break down our starch into lots of little glucose molecules. And then a fat molecule is made up of fatty acid and glycerol. So our lipase is an enzyme that breaks down our fat into fatty acids and glycerol. So each of these are your big long chain molecules getting broken up into smaller pieces, so our degradation reactions. So obviously we've said there are two types of reactions. So the ones that you need to know are your breakdown reactions, which are degradation, are catalase, lipase, pepsin, and amylase. And there is only one build-up reaction that you need to know, which is really important, and that's a synthesis reaction, obviously, and that would be catalyzed by phosphorylase. So let's go into them in more detail. So obviously your larger molecules changing into smaller molecules, so your substrate is really large and then it gets broken down into a smaller product. That would be called a degradation reaction. So you could draw a picture of the set 1, 2 and 3. So you've got your active site, your substrate fits into your active site, your reaction takes place, and then your smaller product leaves the active site. So amylase is the first one that you need to know, so it's found in your saliva. So amylase speeds up the breakdown of starch into maltose. It can also break down the starch into glucose. So you want to write this down. So starch and amylase give you maltose sugar. So a way to remember it is SAM. So you might want to print out, there's a table in the consumables which summarizes all of the different breakdown, degradation and build up synthesis reactions. So we've got to remember SAM, starch, with the enzyme amylase goes to maltose. So amylase speeds up the breakdown of starch to maltose. So the diagram below shows an investigation into the digestion of starch by amylase using a model gut. Which line in the table below describes correctly the content of the model gut, which should be used as a control? So in a control, we always want to have our water solution, and we'd want to have our enzyme, and we'd want to have no amylase solution. So it would have to be letter A. So we've got our starch solution and we've got our water, but we've not got any amylase. So that proves that the amylase would be having the effect. So remember, the reason for a control is always to prove that something is having an effect. So in this case, it proves that amylase is causing the effect and it's not happening on its own. So the next enzyme we need to look at is catalase. So catalase is an enzyme involved in the breakdown of hydrogen peroxide into water and oxygen. So this is another degradation reaction. So you've got your hydrogen peroxide, which is a long chain, and then it goes to oxygen and water, and this reaction would happen on its own, but it'd be really slow. So catalase, the enzyme, speeds it up. You'll notice most of your enzymes end in A's. So a way to remember this is either HP-Cal, so hydrogen peroxide, with the enzyme catalase goes to oxygen and water. So hydrogen peroxide is obviously your substrate, catalase is your enzyme, and oxygen and water would be your substrate. Or you can remember it, which is my favourite way, is Harry Potter's cat went out. So hydrogen peroxide catalase, and then water and oxygen. So then another degradation reaction that you need to know is pepsin. So proteins are one of the main food groups in your diet, and you need to break them down. So pepsin is one of the main enzymes found in your stomach. So pepsin in the stomach breaks down proteins into amino acids. So your pepsin speeds up the reaction of proteins going to amino acids. Proteins is your substrate, amino acids will be your product, and pepsin is your enzyme. And then you can remember that by PPA. So way to remember it is 2 P's give 2 A's. So protein, and then the enzyme pepsin gives you amino acids. So the diagram below represents a degradation reaction involving an enzyme, which letter identifies the substrate. So if we're looking for a substrate, we're wanting to look at the one that is going into the enzyme, which is A. So this would be your substrate here. And then we would have, if you were asked to identify the active site, it would be the bit where the substrate binds. So it would be this area here on D. And then B and C would be your product. And obviously it's a degradation reaction because it's getting broken down. So we've looked at a lot of degradation reactions, so where the substrate becomes smaller. So this time we're looking at the opposite. So other enzymes do the opposite. So small molecules are joined together to make larger molecules. So this is called your build-up reaction, so your synthesis reaction. So building things up. And we're going to look at the one enzyme that you need to know, which is phosphorylase. So this is a diagram which shows a synthesis reaction. So you see your substrate coming in, so into the active site, and then you see your enzyme substrate complex, and then you see your product, and your product is bigger than your substrate. So it's a build-up reaction. So phosphorylase is found in potatoes. So if you grind down potatoes, you'll be able to get phosphorylase. So lots of plants store glucose made in photosynthesis as starch. So phosphorylase joins your glucose 1-phosphate molecules together to form starch molecules. So you need to know that glucose 1-phosphate is your substrate, phosphorylase is your enzyme which speeds up the reaction, and starch is your product. So phosphorylase speeds up glucose 1-phosphate combining together to make starch. So it's really important that you know this one synthesis reaction and your 4 degradation reactions. So you might want to make it into a poster and then try to learn it by looking at the poster and then trying to copy the poster without seeing the poster a few times. So you need to know your degradation reactions and you need to know your one synthesis reaction. So the 4 degradation reactions that you need to know that where enzymes are involved is you need to know the one involving pepsin, the one involving catalase, and the one involving amylase and lipase. For your synthesis reaction enzymes, you only need to know phosphorylase. So we have a table to summarize this. So here are the degradation breakdown enzymes you need to know with their substrates and their product. So your enzyme catalase catalyzes the reaction of hydrogen peroxide, which is your substrate, to oxygen and water, which are your products. Then you've got your enzyme pepsin, which catalyzes your reaction of your substrate protein, which breaks it down into your product of amino acid. And then you've got lipase, which catalyzes your substrate fat and breaks it down to fatty acids and glycerol. You can remember lipase by liposuction, which usually sucks fat out of you. And then amylase, which catalyzes reaction of breaking down starch into maltose, which we were meant to remember as SAM. So we need to know these degradation reactions, so you need to look over them, write them out a few times, and know what enzymes involved with what substrate and what the products are. The last thing that we need to know reaction-wise is our synthesis reaction, and we know that our enzyme phosphorylase, which is found in potatoes, can catalyze the substrate glucose 1-phosphate being built up into starch, which is the product. So what we would usually get you to do in class is use a plasticine model to create an enzyme with an active site, and you would model your synthesis reaction and then you'd model your degradation reaction. So you'd want to first of all show your substrate, and then you'd want to show your enzyme separate, and then you'd want to show your enzyme and substrate complex, and then you'd want to show your products at the end. So make a plasticine model, or either a build-up synthesis reaction or a breakdown degradation reaction. You must label your model with the following parts. So active site, substrate, product, enzymes. Now you can also do your National 5 textbook biology work, page 38. You can use that for help, and then you can also do the questions on that. So our diagram that we have drawn should look a bit like this. So you've got your enzyme and your substrate, and then you've got your enzyme substrate complex, which is like complementary shape. So your substrate complementary shape to your enzyme's active site. Remember, you have to mention substrate complementary shape to enzyme active site. And then for the final thing, you've got your product with your enzyme. So the something is the chemical that the enzyme acts on. The something is the end chemical produced. The something of the enzyme is where the chemical reaction occurs, and it is said to be something to the substrate. The enzyme and the substrate something match each other exactly. Enzymes are something as only one enzyme acts on only one substrate. So we would want the substrate is the chemical that the enzyme acts on. The product is the end chemical produced, and the active site of the enzyme is where the chemical reaction occurs, and it is said to be complementary to the substrate. The enzyme and the substrate shape match each other exactly, and enzymes are specific as only one enzyme acts on only one substrate. So well done if you got all them answers correct, and you want to make sure you have a note on this before you move on to the next slide. So now you can go into the comprehension practice, so that's in your activities and consumables in 4.2 enzyme structure. And you can try the question free, and then you need to look at the answers and mark it. It's quite a difficult question, but it's quite interesting, and it's quite good to understand what the SQA are looking for when you have to read long passages. So the practical use of enzymes, this is more National 4 than National 5 work, but it's good to know anyway. So an enzyme called rennet is used to clot proteins in milk, which is used in cheese making. And different enzymes such as proteases, etc., help to remove stains in biological detergents. And that's why they can speed up the chemical reactions, and you can use them at lower temperatures, so it's good for the environment. So we've got a cellulase, which helps to digest fruit pulp, which has helped in juice production. You've got papain, which is used to break down tough meats and fibres, which is in meat tenderization. And we've got lactase, which removes lactose sugar from milk, so it gives you lactose-free milk. So from that, you should be able to answer what enzymes and what uses they have. So practical uses of enzymes. So cheese making would be your rennet. Your meat tenderizing would be your papain. Your lactose-free milk would be your lactase. Your fruit juice production would be your cellulase, and your biological detergent would be your protease. So well done if you got that all correct. So common questions that come up in the National 5 biology course about enzymes are as follows. So enzymes are biological catalysts. Diagram below shows part of an enzyme controlled reaction. So you have your substrate and your enzyme, and you've got your enzyme substrate complex. It says describe the features of an enzyme which would allow it to combine with only one substrate. Now this is worth 2 marks. So you need to say 2 things for 2 marks. So you're going to pause the video and you're going to try and answer this question on your own. So the answer to this question would be, the first point would be to say that the enzyme has an active site. Okay, your second mark would be to say that your active site is a complementary shape to your substrate. So first mark saying enzyme has an active site, second mark saying the enzyme active site is a complementary shape to your substrate. So here's the marking scheme. So it says enzyme has an active site, and it must be clear that the active site is on the enzyme. And the second mark would be your enzyme's active site is complementary or a matching shape to your substrate. So for the next question, it says complete the following word equation for the enzyme catalase. Now this could be for any of your degradation reactions, so lipase, pepsin, amylase, phosphorylase, and so forth. So we've got our substrate, which is hydrogen peroxide, and then we've got our enzyme, which is catalase. Then our products would be, and you need to mention what your products would be. So Harry Potter's cat went out, so our products would be water and oxygen. So you've got to look over your other degradation reactions and your synthesis reaction and make sure you know them. So for this question, it says the diagram represents stages in an enzyme controlled reaction. So you've got your first stage where your substrates bind into your enzyme, and then you've got your enzyme substrate complex, and then you've got your product leaving your enzyme. So enzymes are involved in two types of reaction. Identify the type of reaction shown in the diagram above, and B, describe the events occurring in the enzyme reaction shown. So for A, the enzymes are involved in two types of reaction. I'll give you the answers just now. So if you're wanting to answer it by yourself, you need to pause the video. So the answers are that there's two types of enzyme reactions that the enzymes are involved in. It's the degradation and the synthesis. So we need to look at what is going on. So our product is quite big, and then our substrate is quite big, and then our product has been broken down. So that means that it's a degradation reaction. So your first answer is going to be degradation reaction. Then it says describe the events occurring in the enzyme reaction shown. So you want to see what's happening in stage 1. So you need to say that your substrate is binding to the active site of your enzyme. Now it's important that you say your substrate is binding to your active site of your enzyme. And then you want to say that your active site and your substrate are complementary to each other. And you want to say that your substrate and your enzyme complex is then formed in stage 2. And then for stage 3, you're wanting to say that your substrate has now been broken down into smaller components or smaller substances and has been released. So that would be your 3 marks. So for the marking scheme, you've got degradation breakdown. Try to make sure you use the word degradation. And then for the second part, you would want to write, first of all, that your enzyme and substrate join together. And then you've got your enzyme substrate complex, and your reaction occurs at your active site, or your enzyme has a active site, is for 1 mark. You want to say your enzyme and substrate are complementary, specific to each other. So the active site of your enzyme is complementary to your substrate, and they bind together to form the enzyme substrate complex, would give you 2 marks. And then you want to say that your 2 smaller products are made and released. Just what we said before. So you need to be able to describe the importance of enzymes and give examples of reactions in which they are involved. So you basically need to be able to say why enzymes are specific, so that your enzymes have your active site, and the active site is a complementary shape to your substrate, which means that only one substrate will react with one enzyme. And then you also want to say that enzymes are involved in 2 types of reactions, so degradation and synthesis. You need to be able to know your degradation reactions of your lipase, your amylase, like your protease and your pepsin. And you need to know your synthesis reaction of your phosphorylase. And you need to be able to draw your enzyme reactions, so your synthesis reactions and your degradation reactions. So if you're able to do all of that, you can move on to video 9. If you're not able to do that, you need to go back and watch this tutorial video again. Okay, that is the end of this video tutorial. Woohoo!